Hi everybody, welcome back to your channel. And now today we are going to continue with our previous lecture. So if you have watched my previous lecture, then you know that in previous lecture we have discussed the histology of alimentary canal. So let us some brief discussion what we have learned in the previous videos. So we already know that in the histology of alimentary canal, the wall of the alimentary canal is made up of four different layers. The innermost layer is the mucosa, and after that submucosa, then muscular externa, and the last layer, the outer layer part, is known as serosa and adventitia. So this lecture is very important, and also if you watch the previous videos, then it is very helpful to understand this lecture, because that lecture is this lecture is the part of the previous lecture. So let us start. Now in last lecture we have already studied the different layers of gastrointestinal system. Different layers of gastrointestinal system, that is the alimentary canal system. In the previous study, that we have four layers: mucosa, and after that submucosa and muscular externa, and the last layer is the serosa and adventitia. And we know that in the mucosa is again divided into three different layers, and that is lined by the epithelium and lamina propria and muscularis mucosae. So in mucosa there is a three different layer, that is epithelium, lamina propria, and third one is the muscularis mucosa. So here we will focus on the function of epithelial cell. So what are the special features of epithelial cell? What are the characteristics of epithelial cell? So the function of epithelial cell is actually they take a role. They are implicated in protective part, absorption part, and the secretion part. So they perform the role, they protect, they absorb, and they secretion. So let us talk about the first part, that is protective part. So this is your layer of epithelial cells. As we know that these epithelial cells are columnar epithelial cells. Simple columnar epithelial cells, that is the lining the innermost layer of your gastrointestinal system. Actually this epithelial cell, the function of this epithelial cell, that these cells are so specialized and so arranged that if any foreign particle from our mouth, from our gut, if any foreign particle is coming, it does not allow to enter inside our wall of the gastrointestinal system, in the wall of the alimentary canal. So this epithelial cells is protective in nature. It helps to form the barrier that these antigen may be virus, bacteria, or some other antigens, that it cannot enter your GIT system, gastrointestinal system. And then after that, suppose if any bacteria entered your GIT system, it goes to the second wall, and after that the circulation, then it will go to your circulatory system. So this epithelial cell is protective in nature. They do not allow the foreign antigen to enter inside your wall of the gastrointestinal tract. So it protects from bacteria, from foreign antigen or some foreign noxious compounds, noxious element. It protects. This is the role of epithelial cell. The first part is the role of protective epithelial cell. In between the epithelial cell, there are tight junction. So even in between the epithelial cell, there is a tight junction. This is a tight junction, that even from here also no antigen, no bacteria can pass. They cannot pass from this region and go to the other region. So there is a tight junction. So by this way, this epithelial lining, that is first innermost lining of the gastrointestinal tract, protect from the outer bacteria, some noxious element which is trying to infiltrate inside your wall of the GIT. So these epithelials are protective in nature, and this epithelial cell is present here in the mucosa of the epithelial layers. Now come to the second point, is the absorption. Actually, the absorption, these epithelial cells having very important function, that these epithelial cells having microvilli on their surface, microvilli, that is called also brush-bordered. So this microvilli, brush-bordered villi, this microvilli help in the absorption of food, and the role of this microvilli to increase the surface area, that absorption will increase. So this is actually the microvilli is present on your epithelial cells. Now again, the most important point, once again the second most important point, is that presence of villi. Actually villi, what is the villi? Actually this is your lamina propria part. This is your lamina propria, this epithelial cell. This lamina propria part, actually they form the finger-like projection, protrude outside, finger-like projection, and this lamina propria finger-like projection again increase the surface area for the absorption. See, these are the microvilli. As you know, the microvilli increase the surface area which is present on the epithelial cell, and the second protrusion, second area, this fold, this projection outside, this projection again increase the surface area, and that the bigger projection in the lamina propria just below the submucosal part, that is called villi, and this area is called villi. So villi again increase your surface area for absorption. So in the epithelial cell you have the microvilli and the lamina propria. Second is the villi, which both contribute significant contribution in the absorption of your food, just like carbohydrates, lipids, amino acid, minerals, vitamins, water, electrolyte, everything. It helps in the absorption. Now again, sometimes what happens, this epithelial cell secrete an enzyme also, secrete a glycoproteinous material. This is the glycoproteinous material which is called glycocalyx. This glycocalyx is rich in the enzymes. Enzymes are dipeptidase, disaccharidase, which can break into disaccharide into monosaccharide, into dipeptide into monopeptidase. So this glycocalyx is the glycoprotein which is secreted by the epithelial cells, and this glycoprotein, that is called glycocalyx, having the enzymes, that is dipeptidase and disaccharidase, help in the breakdown of the disaccharide into monosaccharide and dipeptide into peptides. This help in the absorption function. Third, in the submucosa part, you will see there is a circular oblique muscles is present. This small intestine here, it is just like it is also a fold, small intestine. This fold in the submucosa is called plica circularis. This again fold is called oblique type of fold, that is called plica circularis. So now we understand the protective roles, functions, and second function is the absorption function. Now come to the secretion function. Now the specialization of this epithelial cell, special features, special functions of these epithelial cells. You know, this epithelial cell, which is the innermost wall of the GIT system, innermost wall of your GIT system. These epithelial cells, they fold and form the gland. These epithelial cells, they fold and form the gland. So this epithelial cell, the gland extend up to the lamina propria, and this is called mucus gland. This is called mucus gland. Second gland, which this epithelial cell again fold and form the second gland, and this is gland up to the submucosa, and this gland is called submucosal gland. The important point here to the submucosal gland is that this submucosal gland is also called Brunner's gland. Brunner's gland. This gland is very important, Brunner's gland, and the role of this Brunner gland is that this Brunner gland secretes alkaline media, alkaline fluid. Actually, what happens when you eat the food, then food goes to the stomach area, it forms the chyme, and that chyme actually is the partial digested food mixed with the HCl, acidic fluid. So this chyme actually coming from stomach, chyme which is coming from stomach has been neutralized into the duodenum, neutralized by the Brunner's gland, which is secreting alkaline substance, alkaline fluid. So these Brunner glands help into the neutralization of the acidic chyme. So it helps to neutralize the acidic medium. So this is a very important point. There is a Brunner's gland which is present in the submucosal gland. This epithelial cell extend to the submucosal gland. The second gland formation is called Brunner's gland. So this point is very, very important. The Brunner gland are the gland which neutralize the acidic chyme which is coming from the stomach, going to the duodenum, going to the intestine, and this Brunner's gland secrete the alkaline medium, and here it is neutralized. Third gland is actually what? They passes all the GIT system. They passes all wall of the gastrointestinal system. That this fold actually extend to the extend from serosa. It passes from the muscularis mucosa, submucosa, even your muscularis external and serosa. So this is called extramural gland. Mural matlab wall. That above the wall, it crosses all the wall, and that forms the extramural gland. Actually, in our body system, the extramural glands are the liver and the pancreas is the best example for the extramural gland. Their ducts are so long that they secrete the enzymes and everything to the duodenum by the extramural gland. So this is the secretion method. This is the secretion, and there are the three glands which is present. The first gland is lamina propria, which epithelial fold and form the gland to the lamina propria, that is called mucous gland, and after that second gland extend up to the submucosa, and this gland having the called Brunner's gland which secrete the alkaline media, and third gland is called extramural gland. So these are the important function, special feature of epithelial cell, that is protective, absorption and secretion. Now come to the lamina propria, specialization of lamina propria. So what is the lamina propria? In the previous video, in the previous lecture, I have already discussed that lamina propria having the blood vessels, glands and lymphatic system, lymphoid tissue, lymphatic system and the nerve plexus. So here the lamina propria. So the lamina propria of the oesophagus, this is the oesophagus. The lamina propria of the oesophagus, the wall of the lamina propria, it forms one fold, and this fold is actually built like a gland, and this fold, this protrusion outside of the lamina propria, this is called the cardiac gland. So in the oesophagus, there is one gland is present which is the fold of the lamina propria area, and this is called oesophageal cardiac gland. Oesophageal cardiac gland. Now come to the stomach area. Now stomach area, this fold extend like a finger-like projection. This finger-like projection, this is called finger, and this is gland. Actually this is the stomach, so this is called gastric gland, stomach gland, gastric gland, which having the finger-like shape. In this gastric gland, please concentrate on this. In this gastric gland, you have few cells which is called mucus secreting cells. These are the cells which secrete the mucus, and these glands also having the oxyntic cells or parietal cells which secrete the HCl. Actually this HCl is very important because here we have the chief cell which are called digestive enzyme or zymogen cells which secrete pepsinogen and prorenin. So this pepsinogen and prorenin, in the presence of HCl, it convert to the pepsin and it convert to the renin, and it also having the gastric lipases which digest the lipid molecule, So here are the zymogen cell or chief cell, that is called digestive cell, and these are the parietal cell, that is oxyntic cells, which secrete the HCl, and these are the gastric, these called the gastric gland, and here having the mucous secretory gland. So gastric gland having the three type of cell, important three type of cell, that is mucous secreting cells, oxyntic cells which secrete HCl, and the zymogen cell or chief cell, which is the digestive enzyme which is present to digest your carbohydrate, amino acid and proteins and lipid and everything. So these are the chief cells. So this is the lamina propria and the important gland associated with this lamina propria, which is from the finger-like projection. Now again come to the small intestine area. This is small intestine area having very special features, very special, very unique structure and very special features. This area is the section of small intestine. So in small intestine you will see, very important, the lamina propria area of the small intestine. As we know that the outer layer is the epithelial layer, okay, which epithelial layer which having the microvilli, but this lamina propria, this layer lamina propria, as we know that this lamina propria from the submucosa it goes to the finger-like projection and increase the surface area, which is called villi. This is called villi, and villi is actually maximum in the jejunum region. It means that this is the duodenum, this is the jejunum, and this is the ileum. So jejunum area, the villi is maximum as compared to duodenum, as compared to jejunum. So villi maximum in the jejunum area of the small intestine. Now the second part, now the second section which is very important. So this is your lamina propria. Again this is your muscularis mucosa, because after mucosa, submucosa, you having the muscularis mucosa. From muscularis mucosa, your smooth muscle cell protrude, extend toward the this villi, lamina propria. This extend to the lamina propria area, smooth muscle cell. So this form the structure of smooth muscle cells. Second, which is very important, formation of lacteals. Formation of lacteals. What is lacteals? Lacteals is actually lymphatic system. This lymphatic system protrude upwards, and this is just covering your smooth muscle cell. So this is your lymphatic system. So what is this? Is your lymphatic system, which is called lacteals. This is very important system. What is lacteal? Lacteal is your lymphatic system which is present inside your lamina propria. This is your lymphatic system, which is also called lacteals. And these lacteals having the blood vessels. So these are the arteries, these are the capillaries, and these are the venous channel. So these are the arteries, and these capillaries deliver all the important oxygen nutrients, and this is the venous channel which take out your toxic material and just it removes out. So this is your blood vessels, this is your lymphatic system, and this is your smooth muscle cells coming from the muscularis mucosa. So this point is very important, that lymphatic system form the lacteals, lymphatic system, and this is the villi, which is the jejunum maximum present on the jejunum area, and this is the smooth muscle cells which protrude upwards, which go upwards, and this finger-like projection, and this is actually forming the structure inside your lymphatic system. Now again this lamina propria, again it form one fold. This is fold, protrusion. This is fold like glands. It is the glands, and this actually area, this structure which is called crypts of Langerhans. This is also very important, that this area, lamina propria area, form the gland, form the fold, and this fold is called crypts of Langerhans, which is also called COL, crypts of Langerhans, crypts of Langerhans. And this crypts of Langerhans, having the Peynis cell, which is very important cells. This crypts of Langerhans having the Peynis cell, And the role of Pannis cell is actually defensin, having the secrete the defensin and lysozyme. We know that the lysozyme is the enzyme which break the bacterial cell wall. We break the bacterial cell wall with the help of lysozyme. So it kills the bacteria if coming inside your GIT system. And defensin is actually help against it provide the immune system. So this Pannis cell actually defensin and lysozyme. So this is actually very important. Pannis cell is present. So the question is that where the Pannis cell is present? Pannis cell is present inside your crypts of Langerhans. This point is very important. This pouches or this structure fold and this structure is called crypts of Langerhans. And these crypts of Langerhans having the Pannis cell are Pannis cell defensin and lysozymes. Now in this area I have told that some lymphoid tissue is also present. So some diffuse tissues also present, that is plasma cells and some lymphatic system. But sometimes what happens, this lymphoid tissue is so enlarged it forms the nodules. It is a big structure, nodules, and this enlarged nodules of lymphoid tissue is called Peyer's patches or intestinal tonsils. It is called Peyer's patches or intestinal tonsil. So Peyer's patches is the enlargement of lymphoid tissues, that is called Peyer's patches. So your important structure in small intestine is that villi, lacteal system, that is lymphatic system, and this is the gland having the crypts of Langerhans, and this is the Pannis cells. Pannis cells secrete defensin lysozyme, and this is the Peyer's patches is present here. This is the nodules of lymphoid tissues is present here. This is called Peyer's patches, and this is also called intestinal tonsils. This is also called intestinal tonsil. So this is your special features of lamina propria associated with the muscularis mucosa layer. This is the mucosa, and these are the special features, and these are the special cells which is present inside your lamina propria and form the very unique, very special type of structures. Come to the large intestine. So in large intestine is very simple. In large intestine, Peyer's patches is absent. Even the Pannis cell is also absent. Only what is present? Crypt of Langerhans is present, but villi is absent. Lymphoid is present, lymphatic system is present. So in the large intestine, this is the small intestine. In the large intestine, lymphoid is present, lymphatic system is present, crypt of Langerhans is present, but villi is absent. No Peyer's patches and no Pannis cell. So this is the special feature of large intestine. And if you come to the anal area, this is up to the rectum. Anal area, so anal area having the lymphoid tissue, sebaceous gland is present in the anal section, and circumference gland is also present, and some hair follicle is also present in the anal system. So this is the special features, structure, function, या characteristic of lamina propria. Now come to the second section, that is enteric nervous system. As we know that after some mucosa, muscular mucosa, we have muscularis externa region having the circular muscle and the longitudinal muscles. So how these muscles are controlled? As I told you that they are nerve plexus. So they are nerve plexus controlling your enteric nervous system. So we know that central nervous system is the brain and spinal cord, but enteric nervous system is the nervous system present inside your alimentary canal or inside your gastrointestinal tract system, that is called enteric nervous system. So basically two type of plexus is present, two type of nerve plexus is present. One is Orbeck plexus and second one is Meissner nerve plexus. Orbeck nerve plexus is called, actually Orbeck nerve plexus is controlled by your central nervous system, and this Orbeck nerve plexus is present between your longitudinal muscles and circular muscles. You can see here that this longitudinal muscle and circular muscle is controlled by your central nervous system inside your enteric system, that is called enteric nervous system, and this type of plexus, nerve plexus are called orbic plexus or myenteric plexus. So these plexus are very important, so you don't confuse the myenteric. This is the myenteric plexus. The second plexus is called Meissner nerve plexus. Meissner nerve plexus. This Meissner nerve plexus is present between the circular muscle and submucosa muscle. So you need to differentiate between these two nerve plexus. This nerve plexus between the longitudinal circular muscles. This nerve plexus, Meissner nerve plexus, is between the circular and submucosal plexus, submucosal area. And this myenteric plexus is called orbic plexus, and this Meissner nerve plexus is also called submucosal nerve plexus. So orbic nerve plexus, which is the myenteric nerve plexus, present between the lamina longitudinal muscles and the circular muscles, which help in the peristalsis, But here your Meissner nerve plexus regulate the secretion of epithelial mucus, So the important two type of nerve plexus is orbic nerve plexus and the Meissner nerve plexus. Orbic nerve plexus is also called myenteric plexus, and the Meissner nerve plexus is also called your submucosal plexus. You can say also submucosal plexus. So these are the two important plexus, nerve plexus that form the enteric nervous system. So these help in the peristalsis, and this secrete the epithelial mucus, and this help in the movement of your GIT system. The circular muscles help in the mixing, and these longitudinal muscles help in the peristaltic movement. Now, actually in the muscular externa, there are some variation in the different part of your alimentary canal, some variation in the muscularis external part. Basically, in the muscular externa, you're in the mucosa, in the esophagus, you will say esophagus above part actually striated muscle is present, and here the below part is smooth muscle is present. It means that 1/3 part is your striated muscles, 1/3 part is your smooth muscles, and 1/3 part is your mix of striated muscles and smooth muscles. So there is degree of variation, some variation is present in your layer of muscular externa in different part of your gastrointestinal tract. So on the stomach area, we know that two type of muscle is present, longitudinal circular muscles, but additional oblique muscle is also present. Now come into this section is your colon system, large intestine. This section is very important. Colon system. Actually this colon system having the circular muscles, but longitudinal muscles is not present everywhere. So this is the circular muscles, but longitudinal muscle is not present everywhere. It is present very few present here. Suppose it is present here, but it is absent here. So this discontinuous of your smooth muscle cell form it's just like a three ribbons, and these three ribbons is called tinea coli. Actually this ribbons, tinea coli structure, which is smooth muscle is discontinuous, not continuous. It forms the sac like region. See, these are the pouches in colon area. It forms the pouches. It also called saculation, sac formation, small pouches, segmented appearance. You can see segment appearance, and this type of arrangement is called haustra. So this is very important, that what is the tinea coli? Tinea coli just three ribbon like structure, that smooth muscle, longitudinal muscle cell is discontinuous in this area, in the colon region, and this colon region form the fold, saculation formation, and that is called haustra, and this is called haustra. So these are your some variation, some degree of variation in the muscular externa area, in the esophagus, stomach, and in the your colon region. So these are the special feature, specializations of your different type of gastrointestinal tissue of your wall, that function of epithelial cell, that is protective absorption and secretion function. In the lamina propria, you having this formation of gastric glands, cardiac glands, and few glands of crypts of Langerhans, pear patches, large intestine, and Penney's cell. And after that, enteric nervous system, you having the Orbeck plexus and the submucosal Meissner plexus. And finally, and in last but not least, your formation of Haustra in the colon region. So this is our second lecture based on the histology of alimentary canal, and this system I have covered, entire system I covered the histology of alimentary canal. So I would like to thank everybody who is watching my video. Thank you very much, and I request you again and again that please subscribe and please provide me confidence, and you are most welcome. Your comments and suggestions are very important to me. Thank you very much.